Well, Mathieu, thanks for having me today, and welcome to the podcast. Um, so the way I like to start the podcast in general, I like to know a little bit about the person I'm interviewing, and would be great if you can tell us a bit about your journey. Like go back as far as you want. What led you to what you're doing today?
First, thank you for having me. So my background is in studying brain disease. So I did my PhD in Bordeaux. I was studying Parkinson's disease, very much from like a translational point of view, so understanding how we can translate therapies into through animal models and approaching the clinic. And then I decided to do a very deep dive into mechanistic cell biology. So I went to New York for a 5 years postdoc, where I was studying very specifically protein degradation, how this organelle in the cell, the lysosome, which is essentially we could say the garbage truck to some extent, is actually eating proteins and using them and recycling all of that. And then I decided to join the Dementia Research Institute a few years ago. I was part of the lab of Karen Duff studying tauopathies, which is a wide range of disease that encompass Alzheimer's disease, for example, but also all of the frontotemporal dementia. And very recently, I started my own group here at the UK Dementia Research Institute, and the focus on the group is about brain aging. So something that we always say is that aging is the most common risk factor for dementia. However, we don't necessarily understand fully what are the mechanistic underpinnings of that statement. So this is exactly what we're going to explore. How is the brain aging, and how the various cell types in the brain are aging, and how can that be linked to the risk to develop dementia?
Can you tell us a bit more about your research? What are the different avenues that you're exploring? I mean, before we started recording, we were talking about Alzheimer, and that I'm coming from the pharmaceutical industry, and I can't tell you how many phase 3 trials failed because they were targeting the amyloid proteins, and it didn't work. Now we are talking more about inflammation, and inflammation leading to the creation of amyloid. So now it's a new avenue. So I'm obviously not specialized in this, but I've been in the pharmaceutical industry for long enough to know that Alzheimer's disease is one of the holy grail of the industry, and it's definitely also a huge disease burden for aging societies. So love to hear the latest and greatest.
So I think one of the common consensus these days is that first, Alzheimer's is a disease that starts very early compared to when it manifests in clinic. So there is a lot of opportunities in terms of biomarkers development, understanding which are the patients which are at risk to develop the disease, which are the ones that are going to have the disease, and how we can actually monitor the disease process. We need biomarkers for diagnosis, but also for target engagement, which is something that we have been somewhat lacking for many clinical trials. So that's one aspect. The second is that I think Alzheimer's disease, and I think it's probably true for most age-related disease, or especially brain disease, is the clinical label actually encompasses multiple causal track. So you can have Alzheimer's from multiple different reasons, but it's going to manifest as the same disease. So we're facing a large heterogeneity of patients, which also explains why a lot of the clinical trials have been failing, because when you're testing a therapy with a specific mechanistic hypothesis in mind, but you're testing it in a heterogeneous population, you might have only a subset of that population that is actually responding. And this is what all of the kind of post-hoc analysis of the clinical trials have been showing, which is that there is always a small population that respond more than to the treatment than the others. So, but that means also that the way that we're moving forward is it's becoming evident that we'll need polytherapies. And polytherapies that are going to actually target multiple axes of pathology. But my take and my approach is actually less on the treatment aspect, but more on the prevention side, which is kind of like something that we often say here in the Institute is we would like to find a statin for the brain, which is something that is not necessarily going to causally impact the disease itself, but reduce enough the risk to develop the disease later. If we can actually postpone the diagnosis of dementia by 15 years, like it's going to dramatically improve like global health. So my study is very much on this aging, like why most of those diseases needs to happen or actually happening in an age brain. Most of those diseases actually do not appear unless like until you reach like 40, like in the typical familial forms, or 55, 65 in the sporadic forms. So it needs that the brain needs to age for the disease to appear, and that's what we want to understand, with the goal to find something that will postpone the start of the disease.
So going back to one other thing that you were saying, that if you do the post hoc analysis, some patient group were responding better. So do you think that we have potentially a lot of drugs that were potentially efficient if we had a better patient segmentation or inclusion-exclusion criteria in the trials?
I think there is some aspect of therapies, especially like, if you take the example specifically of the anti-amyloid therapies, when we actually go back and we actually look now with kind of like everything that we know now about the newest like antibodies, if we go back to the earlier clinical trials, first we were able to see what went wrong, like in terms of like which species they were targeting, the kinetic of clearance of amyloid, and then you can also do this post-hoc analysis and find that there was actually like traces of improvement in some patients. So I'm not sure that we missed a therapy per se. I don't think so, but I think this aspect of patient heterogeneity is actually like a problem. There's a massive debate right now in the field of Alzheimer's, which is how do you define the disease? And you have two sides. You have the clinical side, which is that you define disease based on symptoms, and then you have the more biology side, which is you define a disease based on mechanism and a tractable biological process. Of course, as a biology, I tend to adhere to one side more than the other, but I think like this is something that is happening right now in the field. The jury is still out there. We don't have yet like biomarkers that are going to predict one-to-one that you're going to have the disease, and yet we also know that the clinical diagnosis of or kind of failing in many instances, or missing diagnosis, or also like confusing some disease for another.
Yeah. I mean, I'm a biologist, so I tend to agree with you as well, because I think there was this paper, I think a year or two ago, that was saying that aging is not linear, and you have two age in life where aging is faster. And I'm 46, so I can tell you it's true, because I think it was 44 and 65. And essentially what they say is that you have a biological process that leads to accumulation of certain things, like misfolded protein, whatever, which leads to aging. And then it's a bit like a glass that fills with water. So the biological process is the filling, and then the disease is when it starts to spill. So when the glass is filling, you don't really feel it. When it's starting to fill, when you turn 44, then this is where you feel it. So I think it's, as you say, it's like the biologist who is looking at the process versus the clinician who is looking at, oh, okay, now I can actually see something. But yeah, going back to what you were saying on the aging process, it's interesting because now I see a lot of pharmaceutical company also that says, I'm not looking at diseases, I'm looking at aging process, because the aging process is the cause of so many of the diseases. So not only Alzheimer, but almost everything. So aging is actually a very it's such an important area of research, but also investment in the pharmaceutical industry at the moment.
Yeah, I agree. Like, so the field of aging about like 10, 15 years ago kind of like came up with the concept of these hallmarks of aging. Yeah. And what's funny about the hallmarks of aging is it's kind of like it's a mixed bag of things, Like you have like DNA damage, epigenetic alteration, many things all going. You have the primary ones, the antagonistic. Anyway, what's fascinating is that if you then try to do the same effort about characterizing everything that we know in every disease, the overlap between the hallmarks of aging and the hallmarks of disease is almost 100%. So it's kind of like mirroring so much. However, I don't think that disease is a continuation of aging. There is like a step somewhere, but we cannot like overlook the component of aging.
So you were talking also about finding a statin, like the statin for aging. So how far are we from that?
Oh, I think we have. Well, you know, I'm like I'm a basic scientist, so my work is really on the mechanistic side of things. Um, there's a lot of effort. I think the field of aging has came up like with a lot of like intervention that we don't necessarily like fully understand. For example, like metformin is one of like the great example. Metformin like affect absolutely everything. We initially thought it was like mostly mitochondrial, but it seems to affect everything. There was all of the rapamycin treatments, and like initially I've been working in the field of autophagy for 15 years, and it was funny because every time someone talked about mTOR inhibition, it was associated with autophagy induction. But then if you go in neuroscience, they're going to tell you that mTOR inhibition, it leads to synapse formation, etc. So what I mean is mTOR is actually like a master integrator of many things, and it's hard to understand exactly what happened when you target it. But all of those interventions seems to to demonstrate that you can affect global cellular process with some kind of improvement. Like how it's going to actually impact disease, we don't know. So, but I would say there is currently some interesting intervention happening, but we're lacking the precise molecular understanding of what's going on.
So you're looking at this from a very, academic, very scientific perspective, obviously. What do you think about, this movement specifically from the Silicon Valley with people like Brian Johnson who are taking, 100 food supplement per day? So most probably in what they take, something has an effect. So yeah, do you have a take on that? And the next question is that as a scientist, do you think that there is something that you can learn from the type of data that we are collecting?
Well, I would, as a scientist, I would say like an experiment with N of one
Yeah.
is like we are not going to be able. I think the benefit for the community of what they've been doing is they've been documenting it a lot, and they are. I think they put some data out there, but I saw like a post on Twitter recently where he was like suggesting that he's been sampling his blood more than anyone else in the world, and there is like a wealth of data that will be interesting. However, how we're going to kind of like make conclusion, I think it's like it's an interesting case report and case study. I don't think we're going to necessarily learn what is a good like aging intervention more than anything like really from it. I think we are learning more from the population side, like more the epidemiologic studies where we see, for example, that how much teaching people how to eat better has been improving health over decades. And like globally, the risk of dementia has been decreasing. The problem is the population is aging, so then the incidence is increasing. But globally, the risk for dementia has been decreasing. And we can attribute that to the fact that people are smoking less, drinking less, eating better, sleeping better, work habits are better, etc. And all of that are kind of like population intervention, which I think we're all going to learn more from that than a single N of one person in the Silicon Valley.
So it's a great segue to the other thing I wanted to discuss with you because so you're talking N of one versus large population. So what sparked your interest in data-driven approaches and AI application in neurosciences?
So I've always been interested in data. I think I've always liked working with computers. No, but like more, what actually interests me is I was taught biology in a very deterministic way. So when I was in like uni, we learned that like DNA contains the gene, then you have the transcript, then you have the protein, then you have the function, and we learn that in a very deterministic kind of linear way. And what we're actually learning more and more is that there's no such thing as like a linear pathway in biology. Everything is like redundant, and there is like feedback loop, and there is pleiotropy of function for most proteins. So and this is where I got interested in, I'm interested in the data, because I think like high dimensional data across multiple scale is going to give us like a better understanding of how those levels of biology are actually interacting with each other, and how does that lead to molecular function. So I would say that I'm very interested in the field of like systems biology, as opposed to kind of like the more typical like mechanistic biology that I was trained on, because understanding systems give us this kind of like more complex understanding and more fine-grained understanding of how like biological processes are happening. So this is kind of my interest in this field that led me to be interested to like data, large like high-dimensional data, multimodal data, and then how to process that, which is like where AI comes into play.
So it's interesting because you said that you were trained in something, and now your interest with data led you to a slightly different direction. So you're younger than me. I also have done my PhD. When did you finish your PhD?
2015.
Okay, I finished it in 2006. So almost 10 years before. And it's true that a lot of the things that I see biologists getting involved in now, they were not trained at all. I think I had biostatistics in my second year of university, and then never had any more biostatistics. And that was the closest thing to number in biology. And now I see, I mean, that's the name of my podcast. It's tech and drugs, and tech, by tech also means the data science side of things. And now I see like such a need for people who speak both language, who really understand the data, understand the science, and are able to bring them together. So as an academic, do you see the way you're training the new scientists, but also the student? I mean, are the curriculum already adapting, or are we still doing some catch up?
I think the curriculum have not been adapting fast enough. I think I had zero lessons about linear algebra, data science. I had a little bit of statistics, but this was very little during even up to end of masters, which is crazy. So that's one thing that I'm telling everyone coming through the lab now, which is that if there is one skill they need to learn, is data science, because absolutely everything that you do, whether you are talking about protein expression or image analysis, everything absolutely is about data analysis, and all of the principles of linear algebra are kind of shared across all of that. So I don't think they're adapting fast enough, but I see. Interestingly, I think this emergence of the newest AI and especially the LLMs is kind of changing everything, and it's making people get becoming more aware of the need for people to understand. So what's funny is that, but maybe we'll discuss that later, is that now there's a new skill that is starting to appear, which is prompt engineering, and understanding how to interact with large language model, which I think is actually necessary. There is no training for it because it's so recent and it's still evolving so fast that you can't get a formal training. But every student, we discuss with students on how to do that, how to interact, how to improve prompts, and you have to build your own kind of experience around it. But I think it's very important to be able to learn how to use those tools in the most efficient way.
So I got in touch with you initially because I've seen a paper that was where you were one of the co-authors that was about validating some of the finding of Kosmos. So Kosmos is how to call it, an AI co-scientist. I think that's the best way I will describe this category of products. I've been absolutely fascinated by the emergence of this type of tools, where it's not just about looking for information and just having a dialogue, but something that actually can create new knowledge. So can you explain in your own word what's Kosmos and how you got involved with the team behind that?
Yeah, so I got involved because of a collaboration here at UCL with Kenneth Harris, who is a professor of quantitative neuroscience, and he knew Sam Rodriguez, who is one of the founders of Future House, then became Edison. And at that time, they were looking for people to beta test their product. So I started to work with them on what was even before Kosmos, which we called Finch at the time, which is now became Edison Analysis. So this was kind of the data scientist. And I got really hooked first because I got really well with the team, but also because it was actually fascinating for me, who is doing most of my days, I would say 70% of my time is actually doing data analysis, omics type of analysis or bioinformatics, seeing that you can actually ask an agent to do, and it's going to do it well. And we're talking about a year ago, meaning that the models compared to what we can do today were really simplistic, and they were already able to do very relevant type of analysis. So we started to work on that, which was a single agent doing a specific task. Let's say you have data, do this analysis for me, it does it and gives you the output. You had also another agent which was doing literature search. You ask a question and then it kind of finds relevant papers. when Kosmos came into play, that was kind of the next iteration of those models. The idea is that behind Kosmos is that they wanted to build a system that will be able to do like the full scientific process on its own. And what I mean by that is formulating an hypothesis, finding papers that support that, analyzing data, and then refine your hypothesis. Doing this kind of like circle between analysis, literature, hypothesis, literature, analysis, and then going back and then iterate over that. And that's precisely what Kosmos is. Kosmos is essentially, the way I think about it is like it's an environment where you have many small agents going to do tasks, and they're going to share knowledge around a world model. And then you synthesize this world model to kind of like build discoveries from it.
So what was your reaction when you used that the first time?
So the very first thing, the funniest thing is that the very first, like one of the very first, I would say the third run that we've done with Kosmos is the one that ended up in the paper. Okay. So that's why it was mind-blowing, because like initially, and I don't have an explanation for why it worked so well this time, we got something interesting. And I can go back on what exactly is this finding and why it was interesting for me. Because one of the key aspects of Kosmos is this autonomous component. So these agents are going to do tasks and then report in this world model, and then the system iterates. Okay, so like is doing kind of like cycles in a way. So then based on the world model, create new tasks to dig further into a specific direction. What was fascinating, so I'm going to explain that in detail and then I'm going to summarize it. The first thing is I use data which were single cell transcriptomic data from the brain. And I specifically asked about why some populations of cells are actually known to be vulnerable to die in the context of disease. And the fascinating thing is that it took the data, subset the data to look into the transcriptome specifically of my cells of interest, map a lot of function, trying to understand what was going on. Just by doing a simple differential expression, notice that one gene was like specifically upregulated, and then downregulated. And then then it says, well, if that gene is going down, the other members of this family will go up. And it generated this hypothesis, tested. So then did the analysis and noticed that actually no, all of the members of this family were actually going down. And then it was, okay, what is this family doing in neurons? Went on doing a literature search, realized that those enzymes are very important to control membrane asymmetry, meaning that the cell, when it's healthy, is actually controlling very well what it's exposing to the outside of the cell. This enzyme on controlling how the cell can flip things through the membrane. So then the hypothesis, well, if the cells are losing the ability to control membrane asymmetry, they're going to apparently expose signal that are going to trigger a response from other cells. So then it went further, which is, well, if I can see that my neurons are exposing this signal to be to be destroyed by other cell types, I will check the other cells. And then it went back to the data, extracted the data from those cells, and then looked whether the cells were actually upregulating the machinery to eat the neurons. And it did. And that was, I think, completely fascinating, because if I summarize that, it went on, did an exploratory analysis, found the gene, proposed an hypothesis, checked it in the literature. Based on that, proposed a new hypothesis, went back to the data, extracted other aspects of the data, and confirmed the hypothesis. So that was the very first, like one of the very first tests that we had with Kosmos. And when I saw that, which was in July, 2025, this was absolutely mind-blowing. And then it was completely crazy, because back in February 2025, I was hosting a seminar in the department where we were talking about what's going on with AI, because this was kind of where the attention aspect in the model came, and the model came like vastly better. And like at the end of the talk, we made predictions with the people in the audience, which is how far do you think we'll get in 6 months or in 2 years or in 5 years? And basically, in July, so 5 months after Feb, we were where we predicted to be 3 years later or 5 years later. So that's how fast it went. And since then, it's been everything is even crazier.
It's fascinating for me to hear that, because I worked in couple of years ago in the pharmaceutical industry, and we had collaboration with AI companies, and then we had scientists in the team. And I remember there was a lot of tension between the AI companies and their engineers and our scientists. And sometimes the AI engineer will make some prediction. So it was mostly in the medchem space, like to predict molecules, create molecules, then send them to the medchems so that they will synthesize them and test them. And the number of time I heard from our medchem, like, I don't like their prediction. My intuition tells me it's not gonna work, and sometimes they didn't want to do that. And hearing from you that you've witnessed a system. I don't like to say thinking because like it's we don't know, and it's maybe not thinking, but I will say thinking just for the sake of simplicity, almost like a scientist, and formulating hypotheses, testing them, looking for the data, refining the hypothesis, and actually something that you think is credible and is actually helping you for your own research. I find this mind blowing. And as you said, also like the timeline. I always refer to AI timelines like cat lives. 1 year of AI time is like 7 years of a normal life. I mean, it goes so fast.
Yeah.
Yeah.
Yeah, no, that's exactly that. And I think recently people coined the term like AI psychosis, which is like where you're completely overwhelmed with everything happening in the AI world. It feels like that these days because in the past few months, it's actually been crazy and completely insane what has been happening with all of the autonomous agents and the OpenClaw and Hermes and and the memory aspect. All of that is actually completely changing the game again.
But in a different scientific domain, in mathematics, I've read some paper from Terence Tao. He's one of the greatest mathematicians at UCLA, who was working also with Google, and he's not the type of guy that will hype things, and he's very kind of, what's the word I'm looking, low-key way was saying, yeah, I mean, it's actually helping find new knowledge and discover new stuff. So when you hear, I mean, you and someone like that saying, well, actually, yes, these are useful tools helping me in my daily research, I think it's quite interesting, and I think it will be a mistake from scientists to just ignore those tools.
Yeah, absolutely, 100%. I think we need to get on board with those technologies because we need to understand realistically they're aspects of my daily life that I don't need to do anymore because agents are faster and better. And I know a lot of things that people are worried is about how to verify what comes out from an agent, how to investigate, kind of assess what's going on. But now all of the agents are kind of fully transparent. Of course the reasoning can be hard to access, but when you ask an agent to do some type of data analysis, you'll get the entire code. You'll get everything, and you can and now you can tweak it well enough so that the code is actually not becoming overly complex or weird. It can actually do a very simple code, extremely well documented, extremely well curated, and then you can simply look into it. And of course, it takes time, but there's a lot of things that I don't need to do anymore.
And I want to talk about that a little bit later. But going back to the paper, and so when you started to work with Kosmos, it generated all those things. So then what happened? So you took the prediction and you tested them. Can you walk us through a bit?
Well, that's
the next steps.
Yeah, so that's something that's still happening because the problem is going back to the lab takes a lot of time.
Now, doing things.
Yes, yeah, absolutely. The lab is always very slow, and as you said, I think this is going to be a bottleneck in the future. But yeah, so about specifically these predictions, we've done two things to kind of confirm it. So one thing is already in the paper, which is we looked for other data sets. So in order to try to validate the hypothesis, we simply took other data sets which were completely independent and almost orthogonal because one was studying mouse brain and human brain, and we managed to find a tractable signal that was similar. So we found across several data sets, we find the same signal and we were able to reproduce it. So that that gives us kind of high confidence that the mechanism that was proposed by Kosmos is actually happening. Now going back into the functional aspect, that are like long experiment, because now we have to specifically implement the model, find the right tools to test it like very specifically. This is still happening, and the jury is still out there, but hopefully like we'll be able to kind of like confirm that precisely.
So that's about Cosmos, and as you said, we are living through this almost Cambrian explosion of AI tools, which means that there are things I'm sure like a few years from now we look at them and we're like, okay, why did this exist? A bit like during the Cambrian where you had all those weird animals that don't exist anymore, but some of them led to what we are today. So that's why among all the noise there are some super interesting stuff. So you mentioned to me also that you're working also with other tools and other collaboration, specifically one that's called CBrain. Can you tell us a bit more about this project?
Yes, so CBrain is an American consortium which is centered around Washington University, and the mission of CBrain has been to build AI tools for the research in dementia. They proposed to build various tools. One is a literature search agent, which will have access to a very well curated kind of data collection of papers about Alzheimer's disease, but also will capture how the field has been reacting to it. So in the field of Alzheimer's, we have a great forum which is called Alz Forum, where people are assessing papers, and then people can comment, and you have really a lot of well-known scientists that participate into that. So this is kind of like a second layer of knowledge around papers, around findings, and all of this AI is going to ingest all of that information to have kind of very well curated information about Alzheimer's. And they're also building more kind of similar tools than what Cosmos is, which is data scientist, more a kind of AI scientist, but not because it's not only about data. And I've been helping to develop and test one of the tools, which is Open Scientist. The paper came out a few weeks ago. And what's very exciting is that this is an entirely free tool, open source. You can run it locally on your laptop if you want to, just with an API or even with local model. And it achieves very high performance, close to top-notch models. And I think this is also exciting because it opens first it democratizes the access to those models, but also it opens the fact that we can deploy those models in trusted research environments. So that's one of the big problems that we have. A lot of the data that are coming from patients are protected, and they have to be protected because you don't want to actually connect the data to the identifiers. So all of those data are stored in airlock system that cannot access outside. But there's a huge benefit of having an AI agent to go over those data, but we need to do it in a very secure way. So those models, such as Open Scientist, actually because they're open source, they can't be audited, they can also be deployed in trusted research environment, meaning that this is going to open new type of analysis and new type of ways to actually process data at scale.
I remember when I was in the lab, so which was many years ago, the way my day started is I would sit in front of a computer, go on PubMed, see if there is anything new, read a few papers, and then plan my day. So you told us a little bit about it, that this thing is changing how you're doing research. So as a neuroscientist, what does it mean to work alongside this kind of AI agents on a day-to-day? How has this impacted your life as a researcher, essentially?
So yeah, many ways. So one thing, for example, as the example that you described precisely, one of the first tasks in the morning, as for me, has always been, I'll come, I'll just get a coffee, and then I'll go through emails, and then one of these emails will be the list of papers that were published yesterday on PubMed and bioRxiv and medRxiv and arXiv, etc. And then you get all of those. So I still read those emails, but now I have an agent that actually search all of these database, knows all of my project, and then give me a prioritized list of which are the paper that I should look. And it's actually so like good. It's like it's striking. It highlight paper outside of my field because they use similar method or they use a similar approach, and if when you're screening papers, in all of that, like this mailing list, you typically see only the title and the authors, and then you have to click to see the abstract. But because the agent is able to actually access all of that information, much easier than more easily than me, it actually extracts papers which are even more relevant. So that is a very simple use case that is actually just changing what's my morning routine. And the second thing is how it's impacting my something that I do often. When I read a paper, often I will find a sentence, or I will find a sentence like, oh, that's interesting. I wonder whether that protein that they're talking about is actually changing in one of my data sets. Or the other way around, which is like, oh, they publish a great data set. I would like to see whether my favorite proteins are changing in this data set. Doing that a year ago involved me going back to my data, looking for this protein, computing the differential expression and the statistics, or downloading the data, understanding how they're structured, and then doing the analysis. Right now, I don't have to do that anymore. I simply ask an agent, fetch the data, do the analysis, and report back. And then instantly, all of these I'm scratching my head ideas, I can explore them at scale. And then once you have this information, you log it back in your your lab notebook, and this is something that you can go back to later. And that, I think, is drastically changing how I work.
I think a lot of the scientists I know, including myself, are a sci-fi fan, and I think the prototypical future scientist is Tony Stark. So I think we've all seen those things where he's in his super lab, super modern, very clean, very well arranged, which doesn't look like a normal lab. Let's be clear. And he's asking his AI, say, hey, I want to do this thing and research this thing. So does this feel a bit like turning into a Tony Stark now that you have that at your fingertip?
Yes, I think so. Yeah, absolutely. And I think something that's pushing in that direction is the system like OpenClaw, and this more autonomous agent where you can interact through a gateway where they are just sitting on your computer. And one of the fantastic examples that I have is someone I work with, who is Manny Korpas at University of Westminster. He's been one of the developer of actually he's the main developer of ClowBio, which is a set of skills specifically for biology. And one of the examples he was showing, so he's really interested in pharmacogenetics. So he has his own genome sequenced. And then the example, he was he was walking on the street, and then used Telegram to ask OpenClaw, find this file on my computer, which is my genome, annotate, look for the SNPs, and predict predict how my pharmacogenetic profile is altered. On its own, the agent find the file where it was located on the computer, matched mapped the genome, extracted the SNPs, ranked the SNPs, and predicted that he had something that linked to cytochrome P450, and that makes him likely to process some drugs in a different way than others. All of that, walking on the in the street, just chatting via Telegram to an agent on this computer, and that is actually doable today. So then that becomes the very Tony Stark feeling where you can which is Jarvis, build that for me. And I think this is just the early days. All of that is what has been happening in the past, I would say 8 weeks or 12 weeks now. This is where we're going now. It's going to explode a lot more. But this ability to interact in a dynamic way with the models, the agents, and have them autonomously complete tasks is game-changing.
So another thing which is super important now, because as you say, it can generate so much information, so much data, so much ideas and results at scale. But we also know that it's really good at coming up with crazy things, at hallucinating. So the topic of verification and trust is becoming even more important, because I think, again, we kind of shift the bottleneck from before it was finding the information, analyzing the information. Now all that is super fast. But then it's producing so much information that this is where the bottleneck is can I trust actually what I have? Can I build a whole research program on a set of stuff that my agent told me, or am I going to to start on things that are actually that I should not trust? So talking about that, how do you deal with that, and how do you check for accuracy and make sure that what you get is things that you can actually trust?
Yeah. So I think there's three aspects here. The first one is code review. Then there is about literature search agent, and then there is about formulating hypotheses based on like the integration of those two things. Code review is getting more and more futile because the accuracy of the agents is absolutely insane. Like there is of course a problem of how you make sure that they behave in one way, but all of that is kind of like an engineering problem. Already when we did a Kosmos paper, we had contractors looking at the code produced with Kosmos, and the accuracy was over like 85% in terms of like how we could simply just take the code, rerun it, and find the same conclusion, or actually like a human rewriting code for the same goal and just matching numerically matching the results. And like again, that was like really high. And this was months ago where we did this benchmark. So I think like the models right now are even better. The second thing is about literature search. So hallucination in literature search has always been a problem. I think it's still a small, a problem to some extent. If you only use the free tier version of ChatGPT or Claude and you ask it to like do a literature search, most likely it's going to hallucinate some references. There's an easy way around that, which is every time it produces an answer, you just reply, verify that those papers exist, and typically it works quite well. However, then this is where the dedicated tools, for example, like Edison tools, are like much better than anyone else in the game, because they are now able to like first they are doing like in context citation. They're also like showing you which figure supports which claim. And I've been using those tools for like months, and I've virtually never seen any like hallucinations. The last problem is more about how do you aggregate those information. So how do you translate kind of like going from data to a claim? And for that, I like a few months ago, I implemented kind of like a hacky way to do it, which was to do like some adversarial attacks on the claims. The way it was working is every time the agent is producing a claim, let's say, oh, I found this pathway or this aspect of the cell to be altered in this data. What I was doing is was taking the claim, taking the data, feeding it to an agent saying, can you confirm that? Because we always worried that agents are slightly sycophantic, they want to agree with you, we wanted to make sure that they will not simply just say, yes, it's true. So what I was doing was actually inverting the claims, which was like, oh, this thing is not, or this thing is, and playing with those. And if you do it like at scale, let's say use like 20 verification agents with the same claim and data, and then you take a majority vote, we've seen that most claims like we were able to actually confirm, like essentially what I mean is we could very easily see the claims which are spurious and the one which are solid.
Will this be built at some point into the tools?
Yeah, so yeah, exactly. So this is actually happening now. So that was my hacky way to do it on locally, just like on my laptop. People have been doing like amazing things. There is this Stanford initiative that built this AI scientist which is called Biomni, and which became a company recently, which is Philo. So what they've I was at this conference a few weeks ago, which is ADPD, so like Alzheimer's disease and Parkinson's disease research, and they were actually showcasing one of the newest product that they were proposing. They created a Biomni version specifically for Alzheimer's disease, but also it came with like a database where agents can log the analysis that they are doing. And then on this database, you have other agents that are going to iteratively go over that and review the claims from other agents. So then you're starting to build like a semi-autonomous system that produce claims, log them somewhere, and then you have other agents that will simply just go and review and say, well, that the method was not right, and so like we should redo it, and then the agent can go back, do it again, etc. So that's one type of approach, and there's many others. So I think MIT came recently with, what was this? concept, the infinity corridor, where again, like essentially the idea is always the same, which is like you create an environment where you have multiple agents interacting, you have some producing claims and some which are going to attack and verify those claims. So these, like what I've been building has been done by other in a much better way at scale, and this is definitely where it's going.
You know, I remember when I was doing research, I think one of the challenge was to keep up with the research, especially if you know a domain where there is a lot of competition. So there are lots of papers, and now there are ways of dealing with that. But I have the impression that for the new generation of scientists, it's not only keeping up with their own research, but also keeping up with the technological development and all the tools. And that even for someone, you know, spending their days looking at that, I feel it's a bit overwhelming. So how do you keep up with all the things now that you need to monitor your own research and the tools that may help you for your own research? I mean,
I don't know. I'm not sure I am actually. Yeah, I think so. I'm trying to split my time. Like I'm trying to just dedicate some time to it. I am very much interested in like agentic AI and how it's like impacting science, how it's going to change science. So I've been dedicating some of my time to trying to understand the literature around it. But it's I agree with you. It's like vastly overwhelming. Of course, you can use agents to summarize, you know, but I feel like then it becomes like a bit of like a...
It's a bit of a loop, like agent to look for the next agents.
Absolutely, exactly. So then it becomes a bit of a like, so yeah, so I think it's definitely like overwhelming today. And I think it's going to get even more overwhelming because something that we can predict now is the process of publication is going to go faster. So we just submitted the paper on Monday. We actually acquired the data in, when was it? July, and we got like the dataset process out of the instrument like mid-September. So between September and today, we managed to actually analyze the data, confirm some of the data, write the paper. And that has been done also because of the help of AI agents. So the processing time between raw data to writing a paper is getting smaller and smaller, which means that they're all going to, I can easily envision that there will be more paper out. I don't know how overall, like we'll be able to assess them and review them and publish them, but I can easily see that what the AI revolution doing is increasing the output of research.
So that's actually that's a fascinating point. I was telling you I worked for many years for publishers. So you know, in the past, like research was taking a lot of time, and then you know the publication process could also take a year or more sometimes. But now, as you're saying, like the research process is being compressed, so the research output is increasing. But in your opinion, has the dissemination of science or the publication process been improved proportionally, or is this now like a key bottleneck as well?
I think that, yeah, this is like a bottleneck completely, and I don't know how it's going to survive what's happening right now. I guess the same is going to evolve. I don't know how, but we're facing like few problems. First is like one of the positive things of the AI scientists, I think, is, as you said, compressing the research time. But I think it's also democratizing a lot of like approaches. So, for example, it's actually impossible I can't envision now like a paper, for example, where you say, oh, I found this gene modulated in that disease. It's impossible. You must do a meta-analysis of every data set out there today. Because this, now with an agent, this is a trivial task. You can set the agent to actually find the data, process them, confirm whether you can see your favorite proteins changing in all of these data sets. This is a must. That is going to be in every paper. But that was historically a task that was some lab doing specifically this task. And so the conclusion, like one of the point there is that the paper are getting more and more complex with more and more data, so they are more and more hard to assess as a reviewer. The big papers, it's like impossible. The length and the depth of the data and the findings it's really overwhelming to assess when you're like an academic reviewer reading the paper and providing an assessment. So that is something where I can see like a lot of problem happening because we're simply lacking time and skills to actually assess all of the papers. And the second problem is that, see, if we're compressing like production, then you need to scale also how this is going to be published or kind of like disseminated. So I'm really in favor of preprints because I think like this is a fast way to access the world and to put data out there and for people to build on it. And then you can have like the critical aspect that is going. But there is now a third kind of component, which is connected to what I was saying before about Biomarkers, which is now we have agents that semi-autonomously produce facts. They find patterns in data and they log it, and then they look and then they go over other data and confirm this pattern. What are we doing with that? This is, I think, valuable information. I think there is a really important, like there is a really high value in deploying those type of agents in genomics database or like any type of omics data set, like a database where they can simply just take data and find patterns around that. The prioritization and the investigation in the lab will have to be determined by a human scientist, but a lot of those findings can be actually produced and logged and put somewhere. Is that a publication? What are we doing with that? Because I think this is valuable information from the community, but I'm not sure how like we're going to manage that.
It's fascinating because what you were talking about that, I was like, so is there a world where our current model of publication becomes like too much publication, like too many publications coming all the time, while at the same time you can think about it where you don't have publication anymore, but you have a continuous production and update of the data in databases or in a system, and you don't think anymore in terms of publication, but you think in terms of constantly updating a certain pool of knowledge that you always tap into.
Yeah, so a lot of people have been suggesting this model, which I like because there is like there is a lot of like strength in logging things in the database, because in terms of search and building relations across like between facts, but that means a complete revolution of how we do science. We've been trained and we are assessed constantly throughout our careers of how many papers we're publishing. Like, whatever people say, at the end of the day, this is like a currency that people are looking at wherever you apply, which is how many papers you publish, where you publish them, how many times are you cited. So what like you're describing is a complete reset of this entire system.
How would you assess research or researchers if we were moving to a model where your contribution is the data and the quality of data that you're producing and the insight derived from your data?
I don't know. The answer is I don't know, and I can tell you this is a very frequent discussion that we have with everyone all the time, and I think no one has the answer so far. But I think it's going into that direction. Whether what we want to see is a model where we would have agents logging facts in database and then human kind of aggregating these facts into like a very big story that would ingest or integrate many of those aspects, meaning that we would have less papers, but paper that will be a bit more like, papers that take like 2 years to produce because it's going to ingest thousands of individual facts. Maybe that's a model, like in a way, maybe that's going to be like beneficial, like to actually have this depth, but I don't know.
So to conclude and looking into the future, so you were saying that you had this conference where you sat down and made prediction that happened to be off just a few months later because it went way faster. So now if I ask you, what are the key trends that you see emerging in biotech and neuroscience, particularly around, how AI is impacting all that. So how do you see the next few months or years unfolding now? And I know most probably it's going to be, off, but how do you think about this right now?
So one of my current obsession and what I think is like where a lot of things are going is about building digital brain for people. The way, like I was watching like one of like a YouTube video, I don't remember who did that, but it was like great. The point that he made is like when you're interacting with an agent, if you're not logging the output of that, this is waste of time. Because every time you're actually working with an agent, you're actually producing some kind of understanding. Like, constantly I use agents to fetch papers, to just like, and then to just like put things together, to review some of my meeting notes, to like prepare like a summary note for people that to disseminate. I went to like 2 meetings, I take my notes, I just ask an agent, okay, prepare a plan, and then I'll just like work on it. All of that is something that needs to be logged because it kind of reflects my thought process. And so that's something where a lot of things are going. I don't know how fast it's going, but there is this aspect of being able to build this concept of a digital brain. For labs, for a scientist and for a lab, I think this becomes very interesting because one of the big problems when you're running a lab is that you have a turnover of people, meaning that you're building a set of skills and you're building a set of like knowledge around skills and experiments and topics, and then the moment those people like continue their career, like you're losing that. So the advantage of a digital brain is, and like interacting with everyone in the lab, is that then it becomes to build a system that will be kind of like a cross-generational memory of a lab. And then you can imagine the system be deployed at various levels. You can have it at the individual researcher, you can have it at the lab level, you can have it at the institute level. And I think there's a very interesting opportunity about how agents are going to impact that. And so the way I typically explain or frame it is like everything happening so far has been about research execution, what I say. So execution meaning that the typical task that we do, which is analyze data, read papers, predict experiment, design experiment, do experiments. A lot of that, it's almost done, data analysis, trivial, literature search, it works very well. Now we can move towards research operation, which is how do you manage scientific project at scale? How do you actually build this, integrate this knowledge and then just aggregate it to produce valuable insights for the community? And just one example is once you have your agent is producing the code to analyze data, right? And then you want to write the paper. I actually did it recently. Just by asking an agent to read my code, it actually produced the best ever materials and methods I've seen because it was 100% precise. It said, you've done data normalization this way with that package, that version. Then you've done this and this and that. You produced you use that mathematical statistical model for analysis, and I think that is very interesting. But it's changing a lot of things. How fast that is going, I don't know. I would say all of that is also possible because of the new ecosystem like Hermes and OpenClaw and all of these kind of semi-autonomous agents. I would say typically if you account for this cat and dog ear compression, I would say by summer it's going to look already different than what we're doing today.
Well, Mathieu, that was fascinating, and I think it's one of the best area of research at the moment. This intersection of life science and tech, and how it's changing how we work. So I really loved the conversation, and thanks for having me today.
Well, thank you so much for having me.